Hello and welcome to today's Hydrophilic Coatings Overview, HYDAK UV Introduction Webinar. Now I'd like to introduce your moderator for today, Jim Morant. Jim, you now have the floor.
David, thank you very much. I appreciate that. And for all of you who are attending today for the webinar on hydrophilic coating and the introduction of the HYDAK UV, welcome. Good afternoon and good morning to our friends on the West Coast. My name is Jim Morant. I'm the President and CEO of Biocoat. And our presenter today is going to be Dr. Bob Hergenrother, who's our Vice President of Research Development and Innovation. And Bob has over 20 years of experience in polymers and development of hydrophilic coatings, and he's going to take you through a lot of information relative to the product and the development process itself. The agenda for today, we'll do a brief overview of Biocoat, which will be quick, and then we'll get to the meat of the presentation, which is really talking about the measuring the effectiveness of hydrophilic coatings and how that is done. Look at the particulate evaluation along with regulatory requirements for coatings. And then we'll get into some more of the detail about the hydrophilic HYDAK UV introduction. This will include information and performance data on lubricity as well as durability. And then we want to end it with a question and answer session so we can answer some of the questions that you may have throughout. And today's presentation will take about 25 to 28 minutes to get through. But just for a brief overview, Biocoat was founded in 1991. It was a 29-year-old company. We were started by a polymer chemist out of Roman Haas named Ellington Beavers. As an organization, we have a global reach with HYDAK being our trademark, and we're located outside of Philadelphia, Pennsylvania. But we ship our products into over 14 countries around the world, and those products are commercialized in over 160 countries around the world. And from a best-in-class performance perspective, Biocoat is really known for its neurovascular, cardiovascular, peripheral, and ophthalmology products that we operate within. But the hydrophilic coatings that we develop and utilize with our partners can be used anywhere in the vascular structure. Not to get too much into the details of the coating itself, but we do operate from a bilaminar platform. And Biocoat is one, if not the only company in the world that offers both a thermal curing system as well as a UV curing system. And what you see on the left is this, essentially how the process is put together. So we have a substrate, and the base coat that goes onto the substrate is a hydrophobic base coat that essentially, in my words, not technical words, but normalizes the substrate and enables us to put a chemical bond for the hydrophilic top coat that comes on after that point. And that is either heat cured or UV cured. The interesting thing about heat cure, of course, is that you can coat both the inner diameter as well as the outer diameter. And for UV curing, of course, that's a line of sight cure, so that is operating really only in the outer diameter dimension of the products that are available. And finally, to wrap it up a little bit about Biocoat itself, as a company, we operate within a vertical integration platform and offer a flexible business model. So as you look at the left-hand side of the screen here, what you see is that we offer you a variety of coating options, which include, as I mentioned, thermal coatings as well as UV coatings. But we also offer a variety of different delivery options, which means that we can do the contract coating in-house here at our Biocoat facility in our 1345-2016 certified facility, or we can actually license the reagents to you and you can do them within your own facility through a process known as a technology transfer. So as a company, we seek to be collaborative with our partners, close in the customer service area, but also offer you flexibility in how you operate your business and how you want to be coating your products. So at this point in time, what I'd like to do is turn it over to Dr. Hergenrother, who's going to take us through the information I shared a bit earlier. So Bob, you want to take it from here?
Sure. Thank you, Jim, and welcome to everyone viewing this webinar. So as Jim mentioned, I'd like to review how hydrophilic coatings are characterized and then introduce Biocoat's newest hydrophilic coating, the HYDAK UV. So first, why do we need lubricity in medical devices? And there's several reasons for this. Main part, of course, is that it improves ease of access of those devices to the site you're being treated. If you can't get to the site of therapy, really the device doesn't do any good. And these hydrophilic coatings are benefit devices in the body that literally go from head to toe. And so again, it improves the device access, but even can actually improve the device functionality, or allowing, say, for a neurovascular application to go further distal than would be because of the friction stack-ups, or say crossing a tough lesion by, again, minimizing the friction up to the point of trying to cross there. The coating can minimize or eliminate trauma on the vessels so that they're not spasming. And all of this goes into then shortening the procedure time, which has benefits for both the patient in terms of time laying on the table, anesthesia, radiation exposure, but then also for the medical staff too, for some of the same reasons. Again, they're wearing lead for these angiography procedures, so the operator fatigue, radiation exposure, and then just freeing up the OR for other uses. So what I'm hoping that you'll get out of this is that you can see that friction reduction is a critical design requirement for medical devices, and it's important that you characterize these in a consistent manner. So how do we go about doing this? And so the first part is an lubricity evaluation, where you're trying to see just how slippery this coating is, as well as the durability of that coating during the time of use. So I've got two examples here of how we at Biocoat do evaluations. The primary one we do is what we call a vertical pinch test or a pad test, where we take two pads and plates that we put squeeze together. For us, we're using 470 grams of force with either a silicone rubber or, if in more aggressive cases, a Delrin pad. That we squeeze that together and then pull the sample up between those pads, measuring the force that it takes to slide up there. We can do this for up to 30, we'll typically do up to 30 cycles, and actually can go up and down through there and measuring that force. The initial friction, that gives you a sense of what that sort of coefficient of friction is. As I said, we're applying 470 grams. You want, best in class ones are below, 20 grams or even 10 grams sometimes. And then looking for on those multiple cycles to be able to have good durability, so that basically that force is not changing over time for those different cycles. What this vertical pinch test does is it does a nice job of comparing coatings with sort of stripping out some of the device geometry and other aspects of it, so that you're kind of looking more just at the coating itself. So that's, for us at Biocoat as a coatings manufacturer, we do use this test a lot. The other way of doing this is to kind of look at the entire device and with its coating in a simulated use model, which is like a tortuous path model, which you can measure the trackability as it's going through some sort of simulated anatomy, which gives you a nice evaluation there. You can do that just either by tactile feel, or even attaching a load cell to that and then measuring the force as it's going along through there. The other aspect of coating characterization is a newer one, which is particulate evaluation. And this has been driven primarily by the regulatory agencies that have been asking for test results and justification, frankly, for those levels of particulates that come from coated devices. Here's the challenge. There are many factors that can contribute to particulate generation, not just the coating. There's the coating, there's how well it adheres to the device. There is also more than just one device in there, as well as all the other parts of going into an operating procedure, such as drapes, wipes, just the general cleanliness of the room. There are standards and guidance documents for coatings. The issue is that none are entirely applicable for giving really good guidance on the particulates generated from medical devices and sort of quantifying and determining what those acceptable levels are. So what do I mean by this? So let's give a few examples here of some of these regulatory guidances. Probably the most well-known is USP 788, which is particulate matter for injections. And it does a very good job of providing instructions for counting the particles once you have your vial of particulates is suspended in a solution. And in fact, it defines limits for those particles in sort of two sizes, greater than 10 microns, as well as in greater than 25 microns. And for containers, it will say that it has certain limits, that you should have less than 6,000 particles greater than 10 microns per container, or for 25 microns or above, less than 600 particles per container. The emphasis here is that per container. It is not designed for a medical device. And so translating what is a per container to a device can be a little bit of a challenging issue. And so And the other part of that is it really lacks any guidance on how to generate the sample from a medical device, or as said, what would constitute that acceptable threshold. Another standard is from AMY, another technical report, evaluation of particulates associated with vascular medical devices. And as I had mentioned, it covers particulates from many sources, not just the coating, so that entire procedure. It does give some description on how to collect particulates, but explicitly states that the literature does not provide discrete thresholds to establish safety limits for particulate loads. And finally, ASTM has a few standards for coatings and medical devices. A lot of them revolving around angiography devices, or in fact, probably the stent systems, where you're looking at how to a pathway defined to show that how the stent is secured to the catheter, as well as it started with some drug-loading balloon vascular systems. So as I said, shows those relevant pathways. It was designed to show that stent securement, but again, does not really provide specific recommendations for the specific test method or acceptance criteria for particulates. And then finally, we get to FDA, who has published several guidance documents on medical devices. Two of them I'll bring up here are on PTCA and GuideWire guidance documents. What's interesting for them is they actually recommend counting 3 different bins of particle sizes. So a greater than 10 micron, greater than 25, and then another one greater than 50 microns, using, as they define it, an appropriate simulated use model, which has to be, our judgment in the medical device world of what that appropriate simulated use model is. And then they'll say, collect those and count them, and then provide an interpretation of that particulate data. Of course, the challenge is they're not really providing what an acceptable criteria is for that. So it basically requires putting it somehow on some comparator devices and putting it in context with other devices around the market, or with your experience or other preclinical models. So let's kind of put some of this into examples here, where what we did at Biocoat is we actually acquired vascular catheter evaluations, 12 different vascular catheters, 6 in the neurovascular microcatheter devices, as well as 6 angioplasty catheters, all from different manufacturers. They were all manufactured at their facilities, as well as final sterile packed devices. What we did is brought them in-house and did a coating and characterization, where we were looking at that lubricity and durability by the vertical pinch that I talked about previously. And then also did a particulate evaluation by using a specified tortuous path and counting the particles that we had collected with LIDA obscuration. And then did some staining of those to visualize that coating afterwards with either a toluidine blue or a Congo red dye. So let's take a look at this. This is for the neurovascular catheters, where we're looking at the lubricity of there. And just to kind of walk you through here, there were 6 different manufacturers and brands of catheters we used. 2 of them we knew were Biocoat customers and had Biocoat coating on them, but the other 4 did not. And so we evaluated all of those, ran this through. We looked at 30 cycles that we were pulling these through this vertical pinch test, and measured both the initial cycles and the 30th cycle. So what you're looking for is low numbers and then not changing much over those 30 cycles. And you can see that actually 5 of the catheters have low friction and also good durability. There was a 6th one that had a start with an initial good friction, but you can see that it started wearing off over time. And not only that, from the catheters, there was variability in that, so indicated by the higher air bars there. So this kind of shows one that maybe the durability is not quite as good. The other ones, though, around 10 grams of force when we're applying 470, that's a really good coefficient of friction. And so these were, as I mentioned, all 510K cleared commercially available devices. So this is how we were evaluating coatings up until the particulate situation came along. But when you add in the particulates, you get a really interesting contrast here, is that on these particle counts, again, these same devices, there is a very large difference in the amount of particles coming off of these devices from the same tortuous pathway, being done by the same operator, tested the same way. And so that there's a 20-fold difference in the amount of particles being generated. The NC6, not surprising since the durability is not as good, probably has a lot of higher particles. But the other 5 also, there was a 10x difference in the amount of particles being generated, all with essentially having the same lubricity and good durability from the friction testing. And so, this, the kind of helps differentiate between different kind of coatings. We're admittedly being very aggressive on this, but kind of shows some of the difference that you see from the coatings and these devices. So that brings me to introducing our HYDAK UV curable coating, which is our newest product offering. As Jim mentioned, we're leveraging our experience and history of excellent performance with our thermal cured systems and applying it to our this new UV curable coating system. And part of the goals for this project was, again, we've had a very good best-in-class lubricity, durability, and particulates as our current HYDAK thermal coatings. One of course, the issues with a thermal cure system, it takes a little longer to cure. And so we wanted to reduce that process time to less than 30 minutes from the coating and the curing, that entire process, less than 30 minutes. As I said, the thermal system can take a little bit longer than that. But also then having it sure that we have compatibility with UV cure equipment that is available or sometimes even installed already at some medical device companies, so that it is that people can rest assured that what we're developing doesn't have to change their investment they've already made on some of their coating equipment. That's being done by these UV flood lamps, which you can see on our prototype coating system here. So just to go a little bit on this hydrophilic coating technology for the HYDAK UV. Again, it's this, as I mentioned, this bilaminar system. And we went through it. We have a hydrophobic base coat that we apply that goes, adheres very nicely to the substrate. What we've done is we've changed out the thermal groups and put in copolymerized with some photoactive monomers. And so that is now part of that base coat polymer. What these do is allow it to bond nicely to the substrate, cross-link to itself, then also be available to bond and grab onto that hydrophilic top coat. The other part of it is then, of course, we have the hydrophilic top coat, which is a combination of photoactive as well as just plain hydrophilic copolymers that we have a mixture of them. We dip coat them on standard techniques, apply it on there, and then when we hit that with UV light, again, cross-links to itself, but then also tenaciously bonds to that underlying substrate. So I mentioned about maintaining that best-in-class performance. Also mentioned that we had done some work with the angioplasty catheter, so I wanted to kind of show that here together. What we did here, similar to the neurovascular catheters, is we looked at 6 different angioplasty catheter brands. One of those actually had Biocoat coating on it. The other 5 did not. Again, put it through its paces on the friction testing. And you can see that there's some large differences here. There's some that are good initial lubricity, but then also good durability. There's other ones, such as the PC4, that have not as good of a durability there. That some of that is wearing off, and some higher variation over time. In contrast and comparison, the UV coating has very similar performance to our thermal coating. That was done on catheter tubing that we kind of weighed into here. So we had good lubricity, very little loss of performance and durability. So the other part of that, of course, is the particulates. And what does that look like? Again, the interesting thing is even though the lubricity data on those various angioplasty catheters looked pretty good, the particulate data sort of tells a slightly different story. And there was, again, sort of two different groups. One was sort of those lower particulates, and one of the ones with the higher variability of the particulates. In this, I'm presenting this as, instead of total number of particles per device, a different way of kind of showing that number of particles per surface area, per centimeter squared. And you can see there that, again, our thermal coating very matched up nicely with what we had with the neurovascular. But then the UV coating has also a very comparable amount of particles being generated from this on the low end. Since this is a new product, want to kind of assure people about the, what is the sterilization effect on here. You can see on that graph on the right, there is the thermal system where weighing into particles there. But then 3 from our HYDAK UV, one with the pre-sterile, but then also after ETO sterilization and even E-beam sterilization to give people options on that. And really no change on those, that we can see that particles are staying pretty consistent over those two different sterilization systems. So in summary, what I hope we kind of showed today was that friction reduction is a critical design requirement for many medical devices, and that there's an interplay between the lubricity, the durability, and the particulate generation from these coatings. And to be able to understand this, you need to have a consistent and I would say comprehensive method of evaluating all 3 of those and looking at the interplay between those. And then, as I said, to kind of mention, with our new UV curable coating, that seems to match up very nicely with our HYDAK thermal coatings that we've had and offer a best-in-class performance. And with that, I will end and ask if there's any questions that are from the audience.
Yeah, David, perhaps you can review the process for asking questions.
Absolutely. So to submit a question, you just want to use the Q&A panel located in the lower right-hand corner of the screen. If for any reason that you're not seeing Q&A over there, there is an icon at the bottom of the screen with 3 dots in it. And if you click on that, you should be able to access the Q&A panel. Just type your question into the text field and hit send, and just keep the drop down as all panelists.
Sounds good. Thank you, David. I have a first question that's come in. I wonder if you can answer this. The question is, can you please provide more detail on what the primary measurement that the FDA looks at to prove coating effectiveness?
Sure, yeah, it's a good question. And, the FDA is obviously looking for the safety of the devices. And so that is why they've been really asking about this particulate generation. And it's really started with the drug-eluting stents and the coating from there, and then kind of continued to expand onto any coating onto a cardiovascular catheter, and is now pretty much expanded onto peripheral and neurovascular devices. The lubricity and durability part are the tactile feel of that and how it performs with the physicians. So it's interesting. You talk to physicians, they talk about how well the low friction it is. With the FDA, they're more concerned with that particle and the safety aspects of the coating.
Okay, thank you. So we have another question that's come in. Can you provide information comparing hydrophilic coatings to other lubricious materials?
Sure, and that's one that you'll see that different materials, they'll say that are low friction, and they are compared to, say, a uncoated polyurethane tubing or things like that. But for instance, some very hydrophobic materials such as Teflon are inherently lubricious. Although I would say that in terms of a hydrophilic coating is probably at least 10 times less friction than a PTFE. There's certainly silicone oils for those, and there's even some additives that can be added into extrusion. But all these are ones that maybe reduce the friction from a sort of an uncoated catheter tubing maybe by 50%. What we're talking about here is bringing that down to 5% or lower from an uncoated device.
Okay. Thank you. Questions come in. Any issues concerning biocompatibility?
That a good question, and that's one that obviously needs to be looked at. We at Biocoat, when we do our coatings, we put them through that ISO 10993 biocompatibility standards as an assurance to our customers, and that is put in the master files. Obviously, the device manufacturer, you need to kind of look at your device in totality. But we're very cognizant of that and want to evaluate all those for those short-term blood contact, putting that in. But I would say no, that there's not the issues of the biocompatibility.
Okay. You had mentioned, Bob, a little bit about the time aspect during the presentation. The questions come in. Historically, thermal coatings have taken longer to cure than UV. Can you comment on the difference between Biocoat's materials and the time for curing?
Sure. So, again, a thermal system, it's sort of one that it gets started and then kind of continues to progress. And there's a time versus temperature relationship there. So, they're not curing at room temperature, but if you heat that up, they'll go faster for that. Obviously, there's an upper limit because the device itself has some, you don't want to kind of overheat the device too much. What we found is that the curing would need several hours. We've actually recently found at Biocoat we can kind of reduce that to about a little less than an hour for our thermal cure systems to have a nice good cure. In contrast with the UV system, when the ultraviolet light is on, it is bonding. When you turn that off, it is not reacting. So it's sort of a exposure to that UV light, but it's much more intense and rapid. And so it is a matter of minutes for each cure cycle. And so when you look at between dipping the catheter in, pulling it out, drying it and curing it, and then as they're showing on ours, the two systems, you can bring that down to well under 30 minutes then at that point.
Perfect, thank you. Questions come up on the range, the UV range required for curing the UV coatings. Can you comment on what we're using in the R&D center?
So the UV range, this is in the UVA range of the ultraviolet spectrum, and then it depends on the lights. With the flood lamps, which I think a lot of people are familiar with, that's where you get these several minutes. There are certainly lights that are more intense that could reduce that even lower, but you need a certain threshold to kind of make sure you're activating things. But in terms of the wavelengths, it is in that UVA range.
Okay, thank you, Bob. Listen, we're at 2:30. We mentioned we're going to be about a half hour for the entire webinar, so we'll take one more question. The question is, are there limitations to the type of substrates you can apply, either the thermal cure or your UV cure systems to?
There are some limits. I would say in general we can coat a lot of things either by directly or with for metals, for instance. Sometimes you need a primer layer. Or there are some plastics that maybe need to have a bit of a plasma or some treatment to kind of make it more amenable to the coating. I will say, for instance, putting a hydrophilic coating on say a PTFE is usually not something I'm in favor of because you've already got sort of an inherently lubricious material, but it's very hydrophobic, putting a hydrophilic one on there. But I would say most of the hydrocarbon polymers that we can do, we can coat. Some are a little trickier than others. But that's where our team, as I said, we stick things to surfaces, and so we'll work with you to optimize a coating process for your device.
All right, thank you, Bob. And I know that there are more questions that are popping up, but we're conscious of the time this afternoon as well. So if you go to the next slide, I'll wrap up. And if you stay online for a minute, we're going to give you a webinar code that you can use to see about getting your first 10 samples coated at Biocoat at no charge. So we'll just wrap up by talking just a bit about the performance here. So what we've tried to do today is to give you an overview of a new product. So in the hydrophilic coating marketplace, there hasn't been a new product out in quite some time. So at Biocoat, we're proud to be able to introduce this new HYDAK UV product. You saw the performance data, but ultimately that performance data needs to take place on your devices with your substrates so you can see the value of it as well. But the proven best-in-class performance is what we wanted to do here today, is to give you a high-level overview of what we're seeing. In addition, the rapid turnaround times with our application development team takes place on a regular basis. We turn your products around within 5 to 7 business days typically. And they're flexible, both pricing and business models, as I had mentioned to you earlier. And one of the things that we pride ourselves in is at Biocoat is a high-touch customer service to support your project. We have teams here of engineers that work directly with you and your engineers and research development teams that make sure you're getting all your questions answered and that your projects are moving forward at the pace that you need them to. So on an overall basis, if you're interested, Bob, if you go to the next slide, if you're interested in testing the coatings, we provided this code for you. If you go to sales@biocoat.com and give an overview of what you're looking at and then put in the code webinar20, the first 10 samples that you send in to us will be done at no charge for you, and we'll do a rapid turnaround time for those as well. So ultimately, we know that every product is different, substrates are different, companies are different in terms of what you're looking for. That's why we're going to provide this for those folks who took the time to be on this webinar with us today. So the first 10 samples will be coded at no charge, and we do a rapid turnaround on those as well. So our ultimate goal is to be able to give you the opportunity to test our products, to try them and see if they work for you. We're confident that with either our thermal cure or our UV cure systems, that will be able to meet your hydrophilic coating needs as you look to commercialize your products and get them to market. So with that, Bob, I'd like to thank you for taking us through this information. I'll say to those people on the phone, if you'd like more information or would like for Bob to do a personal overview of our products to your teams, please get in touch with us again at sales@biocoat.com, and we'd be happy to set up some individual or team conference calls or webinars to go through the data as well and get to know you, your teams, and your products even better. So with that, I'd like to say thank you. I wish you all a happy afternoon, and take care. Thank you very much. Bye-bye.